All right, guys, we are going to talk about mutations, specifically chromosomal mutations today. And then our next flip is going to be on gene mutations. So we are looking at SB2B. So SB2B has us looking at how genetic information is expressed in cells, but we are focusing on the claim that inheritable genetic variations can result from a variety of different things, including new genetic combinations from meiosis, like crossing over in non-disjunction, non-lethal errors that occur during replication, things like insertions, deletions, and substitutions, and or heritable mutations caused by environmental factors, things like radiation, chemicals, and viruses. So you should be able by the end of class to talk about and distinguish between the different types of mutations, explain how genetic errors occur, and explain how errors in the genetic code can result in possible changes to proteins. So remember that when we were talking about transcription and translation, we were talking about the central dogma. We know that a DNA molecule is the code that makes us who we are. It's divided up into smaller units that are called genes, and those genes provide instructions for building proteins. So, the process for how DNA directs the construction of a polypeptide is transcription and translation. Remember, that's the flow of information. We start with information as DNA, and ultimately, we end up eventually at a polypeptide. So, transcription is when the DNA sequence is copied into the mRNA sequence. The mRNA sequence, remember, is small enough to get out of the nucleus. So it leaves the nucleus, goes into the cytoplasm, and meets up with a ribosome where translation occurs. And that's the sequence of mRNA getting decoded into the polypeptide, the amino acid sequence of the polypeptide. And eventually, that polypeptide chain is going to be folded and compressed and manipulated into the right shape to have a final protein. Um, so let's talk about mutations.
From X-Men to Ninja Turtles to the metahumans of Static Shock, science fiction is full of mutants, complete with laser eyes, weather control, and telekinesis. And in these media, mutations seem to be the thing separating the humans, or turtles, from the superhumans, or super turtles. But the truth is, everyone has mutations. These complex genetic changes can be helpful, harmful, or just neutral. And while mutations haven't led to bona fide superheroes yet, there are mutations that have given us some pretty unique abilities, like the ones that let some of us experience the wonders of coffee by metabolizing caffeine more efficiently, or the mutations that let some people feel rested with less sleep than others. But there are also mutations that can cause conditions that shorten people's life expectancy and affect critical bodily functions. So the more we learn about how mutations really happen outside the movies, the more we can help people live healthier lives. Hi, I'm Dr. Sammy, your friendly neighborhood entomologist, and this is Crash Course Biology. I think I left my theme music in my super suit. Has anyone seen my super suit? Where is my suit?
All right, guys. So a mutation is any change to the structure of DNA. Most genetic differences among organisms, like hair color, eye color, skin color, began as some sort of a genetic mutation that was not harmful and eventually became part of the gene pool. Every unique allele began as a mutation of an existing gene. And mutations occur naturally. They can totally occur naturally as accidental changes to the actual DNA level or as accidental changes at the chromosomal level. Some mutations could give a different amino acid sequence within a protein, and that can be problematic. And we will talk about it.
The word mutation comes with a lot of baggage from popular culture. But a mutation is simply a change in an organism's genetic code, alternatively called a variation. You see, DNA contains sequences of nucleotide bases, and each base is represented by a particular letter, A, T, G, or C. Stretches of these sequences of letters make up genes, and genes are like recipes for countless types of proteins that organisms rely on. A mutation occurs when the letters in DNA are altered. This can sometimes change the protein that section of DNA codes for, the way a new ingredient can totally change a dish. Like if you accidentally added sugar instead of salt, which Trust me, will really ruin your date night. But mutations can also happen in DNA that doesn't code for proteins. These mutations occur quite often with no real effect, kind of like the garnish on your plate. And mutations can happen for a variety of reasons. For example, there might be an error during DNA replication, a process that's happening pretty much constantly in the cells in your body. Or an organism might be exposed to a mutagen, a chemical or physical factor that can kickstart changes in DNA, like UV radiation from the sun or the chemicals in tobacco products. Side note, mutagens in fiction create smart-mouthed karate turtles named after Renaissance artists. Mutagens in real life often produce non-functional cells or even cancers. And yeah, that includes nuclear radiation. Though as far as we know, there are probably no radiation-induced Godzilla-sized lizards lurking beneath the Pacific Ocean. The Marianas Trench is pretty deep though, so chances aren't actually zero. Anyway, there are also some mutations that are considered inherited mutations because they can be passed down from parent to offspring. This includes hemophilia, for example, a condition that makes it harder for your blood to clot properly. Now, like life itself, mutations are diverse, and their effects vary widely. But there are two main factors that help to determine the effects of a mutation. The first is location. Different cells in different parts of the body can end up with the same mutation, but the effects are going to look different in cells that create your heart muscles versus cells coding for proteins in your brain. So where a mutation occurs in the body is going to help determine its impact. A mutation that occurs after conception is called a somatic mutation, and it can occur anywhere in the body except the egg or sperm cells. Somatic mutations don't get passed on genetically. But if an organism develops a mutation in one of its egg or sperm cells, well, that altered DNA could be passed on to its offspring cells. This is called a de novo mutation. The second big thing that helps determine a mutation's outcome is the how factor. How did that mutation alter the DNA? One common how is substitution, where one nucleotide is substituted for a different one. Depending on which letter is swapped, this could mean nothing noticeable happens, or it could potentially affect.
All right, guys. So we're going to talk about mutations at the chromosome level today. Those mutations at the chromosome level are ones that happen during meiosis. So during meiosis, remember that in prophase one, our homologous chromosomes, they line up together and crossing over can occur. When crossing over happens, there can be errors that happen in that exchange. There's a few types. There's inversion, there's translocation, there's deletion, and there's duplication. So inversion is when a segment of the chromosome is reversed end to end. So instead of like our original one here being A, B, C, D, E, F, it's now A, E, D, C, B, F. So the B to the E got completely reversed. It's put back in, but it's in the different order. Being in a different order means that the protein is not going to be made in the order it should be. Deletion is exactly what it sounds like. This is where a segment of the chromosome is completely deleted. It's completely cut out and it's gone. No way of recovering it. The size of this deletion could be a small nucleotide. It could be an entire chromosome. It could be a big chunk of a chromosome, like you can see in this picture over here. Duplication, again, just like deletion, it's exactly what it sounds like. Part of the chromosome gets copied and a second one of it is inserted. So like here, we've got our original A, B, C, D, E, F, and then our mutation has A, B, C, D, but then we have two sections that are E, E, and F. So E got duplicated. Again, could be a single nucleotide, or it could be an entire chromosome, or it could be anything in between. Translocation is one where segments of the chromosome break off and attach to the other chromosome. This is where segments of the chromosome are exchanged, but they're not exchanged equally. So in crossing over, they should be exchanged equally so that they're the same size that they need to be. With translocation, they're not crossed over equally, and so they are no longer the correct size when that happens. Non-disjunction is when chromosomes don't separate correctly at all. And what happens with non-disjunction, we know that humans have 46 chromosomes. That's 23 pairs. We know that according to Mendel's law of segregation, the chromosomes separate during meiosis into 4 haploid cells or 4 gametes. If the chromosomes don't separate correctly, that's called non-disjunction. So junction is where things come together. Disjunction is where things separate. Non in front of that, it means it's not doing what it's supposed to, so they're not separating correctly. An example of this is called trisomy 21. This is Down syndrome. An individual with Down syndrome, in either the sperm or the egg, they had two copies of chromosome 21, and then the other gamete has one copy. So when the gametes come together, instead of having two copies like you need, they have three total copies. That's why it's called trisomy, Tri, meaning three, trisomy 21. And you can see here on this karyotype, remember we talked about karyotypes previously. Karyotyping is a great way to tell not just gender, this is a male karyotype, but also to see if there are problems with the chromosomes. Here we can see that there are three copies of chromosome 21.
Changes. There are also chromosomal mutations. Remember that chromosomes are made up of DNA and protein, highly organized, and they have lots of genes on them. The human chromosome number is 46, with 23 from an egg cell and 23 from a sperm cell. This type of fruit fly here, however, has 8 chromosomes, so 4 came from an egg cell and 4 came from a sperm cell. Some examples of chromosomal mutations include duplication, where extra copies of genes are generated, deletion, where some of the genetic material breaks off, inversion, when a broken chromosome segment gets inversed, which means reversed, and put back on the chromosome, or translocation, when a fragment from one chromosome breaks off and attaches to another chromosome. There's more mutations than what we covered, of course, but the idea is that there are many different kinds of changes that can happen. If a mutation is going to happen, we already mentioned that there are especially vulnerable times, such as during DNA replication, but also there are other times too, like during meiosis. In fruit flies and other animals, meiosis makes sperm and egg cells that can have half the number of chromosomes as the organism. However, sometimes those chromosomes don't separate completely. We call this non-disjunction. This can result in an egg or sperm cell that has too many or too few chromosomes. It's possible for a mutation to be passed down to offspring. Consider a protist with a mutation. Many protists reproduce asexually, and when they divide, the daughter cell can inherit the same mutation. A fruit fly, which reproduces sexually, can pass a mutation to its offspring if that mutation is found in the genetic material of the sperm or egg cell. You may wonder why we've been mentioning fruit flies so much. Well, they're awesome, but also it turns out that fruit fly mutations and how they are inherited are frequently studied. We have some links in the video description for more info. These studied mutations can also occur in humans, and we'll give a gene mutation example of a substitution in the
Okay, guys. So remember we talked about sex-linked traits. There are disorders that are found on the X or Y chromosome. Those are disorders that tend to be on the X chromosome. They tend to affect males more often than females. There are also what we call autosomal disorders. So autosomal disorders are ones that are carried on all of the other chromosomes, everything but that last pair, the sex chromosomes. Some examples of autosomal disorders include things like Huntington's disease, achondroplasia, which is dwarfism, polydactyly, that's when you have too many fingers or too many toes, or Marfan syndrome. So autosomal disorders are carried not on the sex chromosomes, but on the autosomes. Males and females are equally as likely to inherit these autosomal disorders. If it's an autosomal dominant, that means only one allele needs to be present for the disorder to show. If it's autosomal recessive, both alleles have to be present for the disorder to show. So just a little bit of recap there with dominant and recessive. If you have any questions, let me know. We will jump back into gene mutations next. So I will be back to talk to you about how they happen during replication. I hope you guys have the very best day, and I will see you guys later. Bye.